of my paper is Emergence of Life and Consciousness, What Does Simpson's Paradox Reveal? My name is Prasanta Bandyopadhyay. The paper is co-authored by Gordon Britton, Ives Itzada, who is a physicist, and two of our students, Laila Moeller and Truman Klumke. So this is the overview of the paper. It's a debate over life and consciousness, and where we'll be applying Simpson's paradox, a very interesting statistical paradox and its different varieties, how it can shed light on this debate over life and consciousness. We'll talk about two types of emergence. One is a weak emergence, another is a strong emergence, and what's its connection with the consciousness. This will be followed by emergence of life, where we'll be applying Simpson's paradox. Revisit the emergence of life with the applications of dynamic case of Simpson paradox, and the third type of emergence, and also we'll be applying the same argument to the debate over life, and end with a conclusion. So the debate over consciousness of life is there is some connection between the two. David Chalmers, who is a very influential philosopher from NYU, thinks there are two types of emergence. Both types of emergence, the weak and the strong, the higher level properties are coming from the lower level properties. However, the difference between the weak and the strong emergence is that in the weak emergence, the higher level properties are completely derivable from the lower level properties, which is absent in the case of strong emergence. And David Chalmers argues that the consciousness is the only strongly emergent properties, which is like what is like to see a red bright light, and that experience is a completely subjective experience, cannot be explained by neuroscience. He distinguishes between two problems of consciousness. One is easy and the hard problems. Easy problem is what can be explained by computations and neural mechanism. But the hard problem of consciousness, as I already mentioned, is like what is like to see a beautiful bird and the kind of feeling, subjective experience we have. Chalmers argues that the hard problems of consciousness cannot be explained by neuroscience. Following the footsteps of David Chalmers, Sarah Walker, who is a physicist, and Paul Davies similarly argues that there are actually two problems of life. One is the easy problem and other is the hard problem. And the easy problem is where metabolism, compartmentalization, all those things occur, which can be completely explained and exhausted by the use of chemistry and physics. However, they suspect, that's the word they use, that science cannot explain the hard problem of consciousness, which has to do with the information first theory, to which I'll reveal very soon. The hard problems of consciousness and life. As I already mentioned, the hard problem stem from trying to explain why and how humans and other organisms have subjective experience. So one of the examples which is very popular in philosophy circle is the Mary example, where Mary, who is a very well-trained theoretical physicist, knows all about wavelengths and kinds of things, but never experienced the light ray, because she spent her life in a dark chamber. Once she comes out from her dark chamber and experiences what is like to see a ray. This subjective experience of what is like to be ray is what Chalmers called the hard problem of consciousness, and he thinks cannot be explained by neuroscience. Connected to that is the easy problem of life and the hard problem of life too. So the easy problem regarding life is that terrestrial biological phenomena such as replication, metabolism, and compartmentalization and so on can be satisfactorily understood in terms of laws of physics and chemistry. However, both Sarah Walker and Paul Davies think the hard problem of life is the problem how information can affect the world. And as I mentioned already, they use the word, they suspect that understanding life, the hard problem, correctly require something fundamentally new laws of physics. So they urge the readers to understand why information is central to unraveling the problem of life, and conjecture further that the hard problem for its full resolution cannot be deduced to known laws of physics and chemistry. And Walker calls their theory the information first theory. So let me put the PowerPoint slide to you to see what we are claiming and what is our contribution using dynamic case of Simpson paradox. Simpson paradox is actually a reversal of the association of direction of the data in the overall population when the data are pulled from the subpopulation or from department. This reversal is known as Simpson paradox. There are at least two versions, static version and dynamic version. Static version is where we see one slot, one particular snapshot of the data, and the dynamic case of Simpson paradox is where we continuously update the table for the data, distribution of the data, and sometimes that uploading and pulling the data generates Simpson paradox, sometimes it disappears Simpson paradox. So by using a dynamic version of Simpson paradox in the emergence of life, we argue two things. These are our two contributions. The SP-based emergence of life falls between easy and hard problems of life, that we'll argue. Second, it also shows, this SP-based emergence of life shows that there's a third type of emergence lying in between weak emergence and the strong emergence. So here's an example of Simpson paradox. It's an imaginary data. We're looking at the acceptance rate of women in the college compared to their acceptance rate of men. Actually, something of that sort occurred in the late 60s, early 70s, the University of California, Berkeley. So what we see that if you look at the data, the rate of acceptance of women in each department is higher than rate of acceptance of men in each department. But when the data are aggregated, when they're pulled together, the reversal occurs. That means the rate of acceptance of men now is higher than rate of acceptance of females. So this reversal is known as Simpson paradox. So we have been working on this problem for more than 20 years. So we have a full-blown formal account of Simpson paradox where we are working on a 2x2 contingency table. All possible combinations have been characterized by our for logical and mathematical formulations. So our formal account in terms of two population, A and B, and the acceptance rate for each department, A1, A2, A1 is the rate of acceptance of A population in department 1. Rate of acceptance of A population in department 2 is designated by A2. Similarly, B1 represents the acceptance rate for B population in department 1, and B2 represents the rate of acceptance of B population in department 2. And then we have an overall acceptance rate for A population and B population, and they are branded as alpha and beta respectively. So here's a formal account of Simpson paradox in a thumbnail. So three definitions: bold C, which consists of three conditions, I'll come back to that, and then theta, and then defining Simpson paradox in terms of condition 1 and condition 2. So the bold C consists of 1, 2, 3. The C1 says the condition A1 is greater than or equal to B1. Second condition C2 says A2 is greater than or equal to B2, and C3 is beta is greater than or equal to alpha. Now theta is a specific condition which connects the rate of acceptance of each population in each department to the overall acceptance rate in terms of beta and alpha. So Simpson paradox arises if two conditions are satisfied. Bold C and theta is greater than 0 must be satisfied. Each condition is necessary, but they jointly constitute sufficient condition for Simpson paradox to be generated. We think the principle of collapsibility is a very important principle which holds the key to understanding Simpson paradox. What the collapsibility principle says in simple words, that whatever holds in the departments of the subpopulation also holds in the overall population. And that collapsibility principle does not hold in the case of Simpson paradox, is what surprises the subjects, and they are surprised. So, as I told you that we have a full formal account of the 2x2 contingency table of Simpson paradox. So we include all possibilities. And so here is one case of Simpson paradox which does not involve causal intuition or causal account, but still is understandable or understood in terms of the formal account we have provided. So here is two bags. Each bag contains a particular number of red marbles, blue marbles of different sizes, and then and then we pour together. We'll pour all of the marbles together and pour into a bigger marble bags to see what's the proportion of red and big marbles and small marbles have. So here is a data table for non-causal case of Simpson paradox. These are exactly the same data as we used in the imaginary case of university example. But instead of female and male, the categorical variable, here we have big marbles and small marbles. And the bag 1 and bag 2 replaces the department 1 and department 2. And red and blue, they represents acceptance and rejection. And we are looking at the rates of red marbles in bag 1. Their rates of acceptance of red marbles, which are big marbles, compared to the rates of acceptance of red marbles, which are smaller. So if you look at each bag, we'll see the rates of red marbles, which are big, are actually rate of red marbles is higher than the rates of red marbles, which are small size. Same thing happens for the bag 2. But when you combine them, we see the rate of acceptance of red marbles, which are big, is actually smaller than rate of acceptance of red marbles, which are small. So this is the case of Simpson paradox, which does not involve any causal intuition because here we cannot and do not say that the bag 1 causes more marbles to be bigger size or small sizes. This example is important because contrary to the famous computer scientist and philosopher Julia Pearl, who contends that all cases of Simpson paradox can be resolved by causal diagram and causal do calculus. So here is a counterexample where there are cases of Simpson paradox which could be causal, which could be non-causal, and we'll be using both causal and non-causal examples of Simpson paradox in our talk. So now, leaving behind Simpson paradox, so we are now talking about competing theories of the origin of life. And there is no other place to talk about Charles Darwin than the Kent itself, because Darwin worked here, lived most of his life, and in 1874, Darwin takes life to begin in, quote, a warm little pond, unquote. Now, there are recent debates revolve around whether the energy or hereditary are the key to life's origin. And metabolism first theory is one of the theory, and RNA world theory is also its competing theory to explain how the life could have emerged in early art. So metabolism first theory affords energy, whereas the RNA world theory champions heredity. So RNA world theorists contain that a cell-replicating RNA, which was capable of catalyzing chemical reactions as well as storing hereditary information, forms the basis of first life. So this is the RNA world theory who are claiming why the RNA world theory is the correct one. Leslie Orgel, who is actually a very powerful supporter of the RNA world theory, unfortunately he died like about 10 or 15 years ago, wrote, quote, "There were a few reasons why we favored RNA over DNA as the originator of the genetic system, even though DNA is now the main repository of hereditary information. One consideration was that the ribonucleotides in RNA are more readily synthesized than are the deoxyribonucleotides in DNA. Moreover, it was easy to envision ways that DNA could evolve from RNA and then, being more stable, take over the RNA's role as the guardian of hereditary," unquote. So here is the metabolism first theorist, who of course opposes the RNA world theory. And according to them, life evolved from a sequence of chemical reactions transformation which achieved a greater level of structural complexity than the available starting materials. And minerals play a very important, significant role in the metabolism first theory. According to the minerals are involved in this process as they can provide protection, support, selection, and catalysis. And the energy transfer is seen as the most important aspect of contributing to life's emergence. So here is the inefficiency objection to the RNA world theories. So Robert Shapiro, again a big, big supporter of the metabolism verse theory. Again, unfortunately, he also died 10-15 years ago. Contends that the RNA nucleotides are difficult to synthesize, so he considered synthesis of ribose on the early earth. Robert Shapiro writes, quote, "The evidence that is currently available does not support the availability of ribose on the prebiotic earth, except perhaps for brief periods of time in low concentration as part of a complex mixture under condition unsuitable for nucleotide synthesis," unquote. Now, similar kind of inefficiency objection raised by Orgel against the metabolism first theory. So Orgel considers the reverse TCA, like Krebs cycle, to investigate the chemical plausibility of the metabolism first theory. While the TCA oxidizes acetyl-CoA to carbon dioxide, the reverse TCA fixes carbon dioxide to acetyl-CoA to make usable carbon compounds. Orgel states that the prebiotic catalysts were not efficient and the existence of side reaction would actually siphon the process of reverse TCA. So this side reaction would siphon off carbons captured in earlier steps of the reverse TCA cycle, reducing the efficiency of the total cycle. So let's sum up what you have done regarding the efficiency, regarding the two theories objecting against one another. So we call it an inefficiency objection. So summing up so far, each theory has raised a similar objection to the other theory. Reactions invoked by any of the theories in early Earth are argued to be inefficient. But life did emerge. So how did life emerge? So assumptions of the inefficiency objection. For each model, we make three assumptions. First, as per thought experiments set out by Orgel or the yields reported by Ricardo et al., each reaction must grow in the ratios reported. The functional molecule is stable enough in its environment so that it does not undergo any appreciable loss. And the third one, the supply of the precursor is not limiting. So here is Orgel is talking about against the Krebs cycle. With an overall yield of only 43% led to Orgel to conclude that the prebiotic reverse TCA is chemically implausible. So here Robert Shapiro argues that with yields of ribose never over 20%, Shapiro would contend that the prebiotic ribose production is also chemically implausible. So here is our dynamical case of Simpson paradox applied to the reverse TCA cycle. So if you look at the data, the growth rates for the functional molecules and the non-functional molecules. By functional molecules, we mean the molecules which contribute to the emergence of life and non-functional molecules which do not. So if you look at the growth rates in both places, reaction, we see the rate of growth rate for the non-functional molecules actually higher than the functional molecules. So definitely functional molecules are locally inefficient. But when they're pulled together, the functional molecules overall growth rates are efficient compared to the non-functional molecules. So then Simpson paradox in the prebiotic production of ribose. So here we have ribose and here's a non-ribose molecules. The ribose are the one which contribute to the emergence of life according to the RNA world theory, and non-ribose molecules do not. So if you look at the growth rates in the subpopulation, we see the non-functional molecules are much more efficient than the functional molecules, which are ribose. But overall, actually, the growth rates for the ribose are higher than the non-functional molecules, non-ribose molecules. So that means even though ribose growth is locally inefficient, but they could be globally efficient. So what lesson we have learned? The lesson from the Simpson paradox is that life could emerge from the accumulation of minor products of sub-reactions while becoming major products globally, contributing to the emergence of life as something which is chemically plausible. So what are its bearings on the easy and hard problems of life and easy and hard problems of consciousness? Inspired by Chalmers' work, Walker and Davis believe that the easy problem regarding life is that the terrestrial biology, such as replication, metabolism, and compartmentalization, can be satisfactorily understood by the current laws of physics and chemistry. However, the hard problem of life, according to Walker and Davis, is the problem of how information can affect the world. They suspect understanding life correctly will require some fundamentally new laws of physics. So they push for the information first theory, where metabolism first theory and Darwin world theory serve as hardwares for the first living system. So for Walker and Davis, emergence of life is to be understood in terms of physics, as it is reduced to a question about how information acquired a causally efficacious role in physical system, unquote. And in biological system, top-down causation provides a way to understand how information can play a role. So Walker discusses the evolution of echolocation in both dolphins and bat to be able to use sonar. The common selection process that offers from the ability to navigate shows the same specific mutation in the DNA of both species. So what does dynamic Simpson paradox shed light on the easy and hard problems of life? So you are going to argue that SP-based emergence of life is neither a case of easy problem of life nor a case of hard problem of life. So SP-based emergence of life does not require replication, metabolism to be central in its account, though it does not deny them. Hence, SP-based emergence of life is not a case of the easy problem. We do not deny that we could know the causal story behind how molecules interact. However, currently we do not know that. In contrast, Walker and Davis conjecture with confidence that the hard problem of life cannot be reduced to the known laws of physics and chemistry. Both agree with the significance of both means they and us agree with the significance of top-down causation in biological system where higher level organization could exert the dynamics of the lower level organization. In our case, SP is an instance of the top-down causation. Once we know that SP occurs in the overall population, then we can immediately tell that both the necessary and sufficient condition for the paradox satisfied. Hence, SP-based emergence of life is not an instance of hard problem. It lies in an intermediate position between easy and hard problems of life. Easy and hard problems of consciousness. The easy problem of consciousness is explained in terms of computation, neural mechanism, and the hard problem of consciousness is explaining the subjective quality of experience. Connected to this easy problem, Chalmers talks about weak emergence, where higher level properties are completely derivable from the lower level properties. And British mathematician Conway's game of life is an example of weak emergence. Strong emergence occurs where higher level properties are not derivable from the lower level properties. And consciousness, according to Chalmers, is a case of strong emergence. So what does the SP-based emergence of life says? Both university and the non-causal marble example are cases of weak emergence. However, the SP-based emergence of life behaves unlike the emergence of consciousness. For Chalmers, there is no causal connection between agents feeling happy, sad, and the occurrence of neural correlates. Unlike consciousness, the SP-based emergence of life is very likely to have a causal story behind its mechanism because of its different interaction between molecules that occur. We do not know it now. Hence, SP-based emergence of life is not a case of strong emergence. Hence, SP-based emergence of life occupies a third intermediate type of emergence. Our contribution. We discuss using Simpson paradox to show how life could have emerged by any of the two competing theories, metabolism first theory or RNA world theory. Walker and Davis argue there are easy and hard problems of life. By using sp-based emergence of life, we argued that it is different from both problems of life. Chalmers distinguishes between weak and strong emergence, again using sp-based emergence of life concepts. We argued that sp-based emergence of life occupies a third intermediate type of emergence. Acknowledgement. The work is partially supported by the National Science Foundation under grant DMR, and here's the grant number. But also we'd like to express our gratitude to the NoRCELVI group for organizing this conference, especially Kathy McGrath and Professor Shuan Jeta for their...
Hello, folks. I thank you a lot for the acceptance of my presentation. The object is the genetic code, and I tell you that I'm working on the opposite of it, the complement of it is called the genetic anticode. It's the anticodons that are written in the tRNAs, not the codons that are on messenger RNAs. We got the inspiration for a model that we are pursuing in the search for the history of the genetic code and for the mechanism of its formation. The inspiration we get is from looking at the ribosome. Inside the ribosome, we have three places there for holding tRNAs inside. That's the tRNA that comes in with an amino acid attached to it. There is an intermediate place which is the peptidyl tRNA that has the chain, the growing chain attached to it, and it will be receiving and exchanging tRNAs with the first one. Then it exits from another side. With this inspiration, we had to imagine how it could have started without the ribosome. And we got the idea that the two tRNAs could hold each other together in a dimer that can pair one tRNA with a complement to it. Then in the middle of the picture here, you have the two tRNAs that are paired through the anticodons. The anticodons are a small triplet that is placed in one fixed place in each tRNA. And we have the full complement of all tRNAs there in the table, where each tRNA will inevitably find some pairs to it. The pairs we make follow some restrictions, that the central base pair is exactly the Watson-Crick type. It's either AU or GC perfect. The lateral base pairs in the triplet, they accept the wobble. The wobble that we use is simply the enipurine pairing with enipyrimidine, so that we can have a simple kind of wobbling, and then we can accept versions of pairs, but following this exact condition of having the central exact base pair. We are following this idea on a search that we are going to demonstrate to you. One of the problems this model or this mechanism initiates, it's the problem that when it is working in a initial biotic situation, those dimers will be working like the singularities that occur in the subatomic particle realm, in the quantum mechanics realm, that we have the partners, which are units of energy or units of objects like this. But when you are in a single pair, there is no direction to follow. The complements to one is also the complement to its complement. Then we have a situation that is called by everybody a situation of simple self-reference. One object refers to its other, which is the complementary object to it, and through the complement, it will get back again to its original situation. Then the self and the other are almost equivalent. And what happens with the birth of life is you can put the next, Savio. What happens with when we get to okay, what happens next is another instance of self-reference. You see, when you have the dimer in the previous slide, it was making peptides or proteins or polymerizations of any kind that could make polymers as products. Then the dimer is a producer, and the product is some kind of polymer that would be available at the time, including some prebiotic amino acids. And the consequence that would lead to the origin of the code would be the association between a kind of product to the producer. When the product associates with the producer, it will make a complex which is called, in molecular biology, is called the nucleoprotein aggregate. And this nucleoprotein aggregate will make the objects that will be able to evolve. The conditions for that is when the association, we call it stable and productive, and it then will resist degradation by being associated, either a proteogene associated with the peptide that make a nucleoprotein aggregate that is stable and protected from environmental challenges. And this thing would work better than it were in that previous state where the tRNAs were exchanging products with some others without the protection from the product. The system that is generated with these two kinds of molecules it's a producer-product system like any other that we know in our everyday life. Any producer will have to receive good feedback and stabilizing feedback and productive feedback from the products or from the consumers of the products so that it will continue and go on. This is what we call evolution before Darwin. It will become Darwinian later. Here at the right, we have a picture that is from a real dimer of tRNAs that was obtained in 1986 by Dino Moras in Belgium. It was the beginning of the electronic imaging of molecules. And they produced dimers of tRNAs that, in this case, they survived purification procedures and stayed together, dimerized. Here we don't see proteins because they were not working on that. But it's demonstrate that the pairs of tRNAs happen and can be obtained. Next slide, then. Then we are applying to this methodology and this hypothesis to the genetic code as it is today. We are showing here the genetic code displayed according to dimers, according to pairs. You see the one below is glycine with the Anticode, which is B, which is a non-A base, and two guanine bases, BGG, is one code for glycine. Its complement is another BCC that can pair with each other nicely, and they both code for glycine. And then following this kind of display, we can put the whole set of anticodes of the tRNAs according to this middle stem or trunk that displays all the pairs that are needed to be encoded in the genetic anticode. There are 8 pairs that compose 16 dimers, 16 units that can dimerize to make the 8 pairs. These 8 pairs correspond to the fully degenerate codes, which is single meaning, then each pair has one meaning in the left side, which is the central purine triplets, paired with a central pyrimidine triplets. Then it's central R with central Y, and this is a straightforward pair in the strictly required way, and the pairs in the lateral positions will be the wobbling, as we told earlier, RY. Then we can start the code from this central stem. All the other triplets and amino acids will be added later. One important result from this way of displaying the code is that we could set apart from all other correspondences the main basal composition of these 4 lower dimers in the central trunk, which is glycine with glycine, serine with serine, alanine with aspartic, and alanine with asparagine. These 4 pairs make the fundamental, stable, and simpler of all codes. We will show in the next slide the conditions for that. You see that these 4 dimers here, they compose amino acids that depend for their formation only on single carbon substrate units to start the pathway of the synthesis of the amino acids. The first is acetyl-CoA pathway that synthesizes the acetate unit, which has 2 carbons only, which is the main part of the glycine molecule. Besides this 2 carbon, which is 1 carboxyl and one CH2, you aminate that in glycine straight away. You don't need to go anywhere else. From this glycine, which has 2 carbons, it gets another single unit of carbon of hydroxymethyl to become serine. Then the glycine becomes serine through addition of another 1 carbon unit, which is a hydroxymethyl. Then you have the first 2 pairs made with addition of single carbons. The serine is transformed into pyruvate, which is the precursor to alanine. The pyruvate derives from serine by dehydratation and deamination. Then the pyruvate is aminated again in a different position to become alanine. Alanine becomes aspartic through addition of another CO2 by the pyruvate carboxylase. Then we have here 1, 2, 3, and 4, 5 amino acids generated from single carbon units. And this is the basal portion of the code that supports everything else. All the other amino acids that are lateral to the left side, you see leucine and phenylalanine, or to the right side, which is lysine, glutamic, arginine. All of these laterals are derived from compositions of the basal units that are in the central pair. The main pathways are derived either from pyruvate, which is the precursor to alanine. Pyruvate gives origin to alanine in that single unit, but it's then doubled into a fused compound of 2 pyruvate molecules that gives origin to leucine. It will also give origin later to valine in the top part of the graph. And another way of obtaining complex molecules from pyruvate is to make sugars from it. Pyruvate is a 3 carbon molecule, which is acidic and keto molecule, but it gives origin to the trioses, which is the 3 carbon sugars, on the way to gluconeogenesis. And the 2 molecules of these trioses make fructose, which will start the next pathway, which is the sugar phosphate pathway of synthesis. That's the pentose phosphate pathway, the hexose monophosphate pathway. These give origins to the aromatic phenylalanine. From phenylalanine, other amino acids will come, like tyrosine, histidine, and tryptophan above. Then we have above of these basal single carbon substrate pathways, we have already 2 other ones. One is doubling of pyruvate to make leucine or valine, and the other is from pyruvate to get the trioses, then via gluconeogenesis and making derivatives from the fructose, all of the sugar phosphate pathways. On the other side, going above from aspartate, the precursor to aspartate is alpha-ketoglutarate. Alpha-ketoglutarate that gives origin to aspartate by plain amination is also the origin of the last of the amino acid synthesis pathways. The oxaloacetate that gives origin to ASP unites with acetyl-CoA, is this pathway number 7, unites with acetyl-CoA and gives origin to the citrate cycle, which is the TCA cycle, from which you can have one compound, which is alpha ketoglutarate, which is the immediate origin of glutamine, of glutamic acid. Glutamic acid is the generator of a large family of other amino acids. Glutamic acid may be circularized in its side chain to become proline. And proline, you have to travel down and to the left to go, and proline substitutes one of the glycines at the triplet BGG. Then proline substitutes a glycine here. But this was a problem for the early students of the genetic code, because you will have a proline substituting a glycine. Glycine is 2 carbon, proline is 5 carbon, and comes from glutamic acid. And it's people that didn't know of these basal set and the mechanisms of generating glutamic would be bothered a lot by this. But then if we segregate this basal part of the trunk of the code, we can understand easily how a derivative from glutamic gave origin to proline in a step which happened in the beginning of the codes with one amino acid that is derived from one of the last pathways. The other problem is that in the code, the alanines in these BAG and BAA positions here, the alanines don't exist in the present code anymore. There is only one remnant of the alanines there in some fungi lineages, in some fungi species. One of these BAG codes that are said to be for alanine here remains there as a variant code, but it is functional and it's selected for. And this gives us an explanation for how these early alanines were substituted by the leucines in this position. Here we finish with the problems that observing the genetic code as it is, we can interpret the quality of this through examination of these biosynthesis pathways. Then biosynthesis derived from 1 carbon units as substrates make the stem and trunk of the code, and the derivatives from duplications of pyruvate, duplications of trioses, or fusions of oxaloacetate with acetyl-CoA come in later. And 2 of these consequences are substitutions from alanine to leu, from glycine to pro, and then we can follow on to the other characters of the code. Then this consistency of the succession of pairs according to the modules is entirely in agreement with each other. The succession of dimers with the succession of amino acid biosynthesis pathways. Next slide. Then we will do a closer examination of these 2 conditions that we see. That the basal set here has, when you look at the most basal of it, which is the glycine duplicate of codes, and the serine with duplicate of codes that are in dark green circles, dark green ellipses. This is This is thought to be a remnant of the mechanism of evolution of specificity of the synthetases. We say that the synthetases at this basal position, which is 2 glycines in a pair, 2 serines in a pair, it means that the synthetase is specific in those cases only for the central base pair, which is a GC. It doesn't matter if it's the G, central G, or the central C. It doesn't matter for any of these two attributions, serine to GA, glycine to GG. It doesn't matter which is a central G or a central C. It means the same amino acid, or the same glycine, or the same serine. Then it means the synthetase is specific only for the central base pair. It doesn't matter if it's one member of the complement or the other member. It means the same thing. Then the synthetase is specific only for the central base pair. The complements have equal meaning. And the remnant of this in the code would have been the tRNA variable long arm, which is seen today in the serine tRNAs. Going to the second pair, which is 2 alanines pairing with 2 aspartics. One is plain aspartic acid, the other is aspartic nitrogenated, which is asparagine. Here, the similarity between members of the pairs occurs laterally. We see here that the synthetase is already specific for the dimer, which is the principal dimer of the triplet, but it accepts transitional variation on the 3' position. There is a central A, central U, nice exact pair, but there is some wobbling in the 3' position, which may be either of the purines coding for ALA, or either of the pyrimidines coding for the aspartic or asparagine. Here we see then that this is one step further in the evolution of synthetase specificity. Here it was only the central GC base pair. Here it is the central AU base pair with accompanied by either another purine together with A or another pyrimidine together with U. This is the second step in the evolution, and we see a remnant of this in the alanine tRNAs, which have the acceptor arm as the main determinant of specificity. Then it doesn't matter which the anticodon is, but the acceptor arm determines that it will be corresponding to alanine, and it shows 3' ambiguity. The same here with the aspartic, with the 3' ambiguity for pyrimidines. When we go out and away from this central basal point, we can already observe that all the synthetases have already specificity for the entire triplet. This is obvious when you see that the UU can be asparagine or asparagine with the 5' G or lysine with 5' Y. The aspartic UC can be aspartic with 5' G or glutamic with 5' Y. Then it means that the third base of the anticodon is already specified. Then here in the central base, which is serine, glycine, alanine, and aspartic, the reference for the specificity is one part of the triplet. Here's the for serines and glycines, you have only the central base pair. For alanines and aspartics, you have the full principal dinucleotide of the triplet as the main determinant, and all the others have the 5' position specified because they have already developed a good complement of amino acids to compose the synthetases. Next slide, then, we have
5 minutes, Romeo, 5 minutes.
Yep. Consequences of this. One consequence is then when we get only those two amino acids in the first part, which is glycine and serine, we see that we have glycine as precursor to the purine ring. The purine ring has the 3 components of glycine inside fully inside in the composition of the purine molecule. All these amino acids, serine and glycine, depend on pterins as cofactors for the biosynthesis, and this is derived from purine. Then the metabolism is fully consistent here. When we get to the second module, which is alanines and aspartins, the aspartic is the core of the pyrimidine molecule. And you see that we only get the nucleic acid backbone, which is a sugar phosphate backbone, only in the next step, when we have already the sugar phosphate pathway coming in. This sugar phosphate pathway gives origin first to phenylalanine, then it goes to tyrosine, and the ribose phosphate backbone coming here. Then we have the situation here that challenges the RNA first hypothesis. We show from this chemistry in the pathways of forming the stuff of biochemistry, forming first amino acids, then purines. Then in the second step, we get pyrimidines in. And only in the third step, we got the sugar phosphate backbone. This means that the beginning was without an RNA. And this challenges the RNA first hypothesis. There's no sugar phosphate backbone available for the first synthesis of that main part of the basal set of amino acids. This is entirely from amino acid biosynthesis pathways. Well, the proposition that I can derive from this is in the next slide, please. No, this is for Sabio. We can skip that. You go ahead, Fabio. This proposition of ours from the dimers forming amino acid synthesis was proposed in 2008. In 2022, it was demonstrated from this paper that is shown here, that you are seeing it is in 2022. It demonstrated with the tRNA acceptor ends making peptide synthesis. But the explanation for the first steps cannot be done with this because there was no RNA there. Then we come to the next slide. Then this is the last slide showing that we can form dimers with polyglycans. You see in green, You see that paper? That's the early polyglycine days. These polyglycins were studied in 1951, 1953 by Linus Pauling when he was describing the secondary structures of proteins. These works of Linus Pauling were the inspiration for Watson and Crick doing the correlative work on DNA structure and the RNA structure. But in those papers of Linus Pauling, they were already showing that polyglycine was able to form sheets and fibers, and these sheets and fibers can make dimers. You see that in this right part of the picture, you have two layers of polyglycins uniting together in a sheet. They call it rippled sheets. It's different from the pleated sheets of plain proteins. It's only polyglycine that can do this because glycine is achiral. Then they can get together. And this is exactly the backbone of the peptide nucleic acid molecule that was proposed by the Danish Nielsen on the early biotic peptide nucleic acid proposition. And then I think we can offer this as an explanation to be explored to imagine how would have been the early days of the genetic anticodes.
Some authors think that originally biological systems only had RNA, so they were structurally solely by RNA. And other authors think that it's actually RNA and proteins working together from the very beginning. So in the past few decades, we have learned a lot about RNAs. We have discovered messenger RNAs, we have discovered transfer RNAs, the very centrality of ribosomes in the whole genome to proteins pipeline. And also have in the past 15 to 20 years, we have discovered a lot about non-coding RNA. So in the very process of cell maintenance. So as we can see, RNA is both important in the modern day for cell maintenance, but also in the very origin of life, because probably it already had a very highlighted spot in life. Initially, RNA probably replicated by the help of ribozymes, and then ribozymes were replaced by RNA-dependent RNA polymerases. This is an in silico prediction of an RNA-dependent RNA polymerase from a virus, just to illustrate it a little bit. The emergency of RNA-dependent RNA polymerases, it was very important because it allowed for the RNA to remain as a main molecule while there was also the synthesis of proteins that could replicate itself, that could replicate the RNA. But then how did RNA transition to DNA? Because nowadays, most lineages are DNA-based, with the exception of RNA viruses. To try and study this, we tried to focus on the enzymes which could be related to the very transition of RNA to DNA. So we focused on DNA-dependent DNA polymerases, which have three main families, which is A, B, and C. While they are not homolog at a level of amino acid sequences, structurally they are quite similar. So this has led some authors to actually say that DNA has been fixated a few times, probably twice in bacteria and then once in archaea/eukarya. So we should note that while structurally they are similar, there is this difference in amino acid sequences. So in this context, we decided to focus on RNA-dependent DNA polymerases because DNA-dependent polymerases, obviously, they can only make DNA from DNA. But then RNA-dependent polymerases, they can make DNA from RNA. So they are probably important in the very transition from RNA to DNA. RNA-dependent DNA polymerases, they are actually also called reverse transcriptases. I'll probably be using both names during this talk. But how does a reverse transcriptase work? Actually, it grabs the single-stranded RNA because it has RNA-dependent DNA polymerase activity, and then it makes a single-stranded complementary DNA. And the interesting thing about these enzymes is that it actually also has a isonuclease catalytic domain. So it can actually destroy this later strand of RNA. And then from the RNA-DNA heteroduplex, it actually forms another complementary DNA strand and then forms a double-stranded DNA. So as you can see, it has both a RNA-dependent DNA polymerase activity and also DNA-dependent DNA polymerase activity. Another work from the same study group that I'm a part of from a few years ago actually studied the natural history of DNA-dependent DNA polymerases, focusing on the B family of this family of enzymes. And they came to the conclusion that actually this specific family came from the RNA-dependent DNA polymerases family. So this work that I'm explaining right now is actually quite similar to this one. So we had the same idea, but with RNA-dependent DNA polymerases. So in this present work, we actually reconstructed an ancestral sequences of RNA-dependent DNA polymerases. We structurally modeled those sequences, and then we made some inference about the possible origins from RNA-dependent RNA polymerases. To get to the actual methodology part, we took 74 RNA-dependent DNA polymerases from the RefSeq database, which is a reference sequence database. And those RNA-dependent DNA polymerases came from archaea, bacteria, and also some viruses. And then we also use an RNA-dependent RNA polymerase as an outgroup. We took all of these sequences and then we aligned them using the MAF version 7 algorithm with default parameters and also homolog analysis, also supplemented by homolog analysis using 600 homologs. The best evolutionary model was determined with Model Finder. The best one was the big parameter with WAG plus F plus R6 model. And the phylogenetic tree was constructed using the maximum likelihood method with 1,000 bootstrap replicates in the IQ-tree algorithm. And the ancestral sequences were then obtained from the MEGA7 algorithm. And we used 3 parameters actually. First, there were sequences with so by parameters, I mean cutoff points. There was a complete deletion cutoff point, a 95% and a 90%. But what does that mean? It means that in the complete deletion parameter, only sites with amino acids in all sequences for ancestral sequence reconstruction were considered. In the 95%, it is required that 95% of the sequence have amino acids at a given position to be considered in the reconstruction. And for the 90%, it's the same thing, except it's 90% and not 95%. So we took all of these ancestral sequences, the complete deletion, the 95%, and the 90%, and we modeled them in iTaster, which is an online program that you insert the amino acid sequence, and it gives back to you the structure, the 3-dimensional structure of your protein. It also can use like some functions and some ligands and some stuff like that. It also can relate to like perhaps the genes that it might be related to. We also use the interpro algorithm to determine functional domains. And finally, we took 39 RNA-dependent RNA polymerases and 10 RNA-dependent DNA polymerases from the protein data bank. And then we align everything using the team align tool. And then we took the resulting RMSDs, which means root mean square deviation, which is a measure of the average distance between the atoms of the superimposed molecules. And then we took all of this data and inserted it into a distance matrix, which we made using the TREX online algorithm, which is a web server for phylogenetic trees and networks. The tree reconstruction method used was the weighted least squares method, NW. with IMW global optimization. So finally getting to our results and discussions. Why did DNA replace RNA anyway? It's probably related to the greater stability of DNA. And DNA is more stable than RNA because thymine is more stable in the uracil, and deoxyriboses are actually more stable than riboses. So this probably worked as a primary evolutionary force during the transitions in the initial states of biological systems. So in general, DNA genomes have a greater stability than RNA genomes. There's a car going by the industry. And so this allowed organisms to have larger genomes. A very good example of this that I like a lot is that DNA viruses are actually a lot bigger than RNA viruses. And all examples that we know of big and giant viruses are actually DNA viruses. So when you're examining nucleic acid polymerases, you can identify several structural similarities in functional domains, which suggests a sequence of diversification processes, which can lead to new functionalities. But among the similarities, one that stands out is the palm domain, which is this one, which is present in a series of classes of proteins. And despite catalyzing different reactions in each of those classes, it's still structurally similar in all of those, and it is present in all types of polymerases. So it is interesting to note that the steps leading to the emergence of a new class of polymerases, and thus the very transitional process from RNA to DNA, may have occurred through small structural alterations in a pre-existing class, just like we argued for the DNA-dependent DNA polymerases from RNA-dependent RNA polymerases. So in this work, given the structural similarities between RNA-dependent RNA polymerases and RNA-dependent DNA polymerases, we actually tried to study and hypothesize if there was some sort of evolutionary process between the two, and perhaps if RNA-dependent DNA polymerase could have come from an ancestor that was more like an RNA-dependent RNA polymerase. So as I already mentioned, we had 5 ancestral sequences reconstructed, one with the complete deletion cutoff, 2 with the 95% cutoff, and 2 with the 90% cutoff. We used the interpro algorithm for motif identification. We actually saw that these ancestral sequences, they have catalytic domains, much like the modern RNA-dependent DNA polymerases. So this data actually demonstrate that despite the diversity in primary sequence, the amino acid sequence, there is still a portion of the protein that remains conserved in the structure of the protein, which may have been inherited from the ancestral groups of RNA-dependent DNA polymerases. This picture actually shows of our models from the ancestral sequences generated by ITASR. When you do in silico prediction of proteins, you have a lot of parameters that you can use. For example, TM scores, C scores, RMSEDs. In here, we decided to focus on C scores, and later on, we'll talk a little bit more about RMSEDs and TM scores. So the C score can come from -2 to 5. So all of the models are between -0.42 and -0.97. So it's actually a very good number. So this means that our models are actually quite reliable. And another interesting thing is that the ITASR, as I mentioned, it can also give some gene ontology information. So all of our models, they have some sort of gene ontology related to RNA-dependent RNA polymerases. And GO scores can go to 0 to 1, so the closer to 1, the better. And all of our models are ranging from 0.62 to 0.83, which is quite reliable as well. So what do all those numbers mean? It means that in terms of primary sequences, our proteins seem to be more related to modern RNA-dependent DNA polymerases, but structurally, they seem to have a greater similarity with RNA-dependent RNA polymerases. It's almost as if they are a transitional stage between ancestors RNA-dependent RNA polymerases and the modern RNA-dependent DNA polymerases. We actually think that we may have captured the transitional stage between those two types of proteins. So to keep up with that hypothesis, we made some other data analysis. We compared the main with modern representatives of RNA-dependent RNA polymerases and RNA-dependent DNA polymerases, and study both RMSD values and TM score values, which are also parameters for like how good your in silico prediction is and stuff like that. We found that the RMSD values, they seem to show that there is a greater structural similarity of our ancestral sequences with RNA-dependent RNA polymerases. But especially when it comes to the catalytic domain. But the TM scores, they don't support this, which seems a little bit contradictory at first. Because the TM scores are related to folding similarities. So it's like they're saying that it's less related to the way that RNA-dependent DNA polymerases fold. But although they seem contradictory, as I said, when you analyze both data and also the previously mentioned data, you can see that actually they seem to reinforce the idea that this is with our ancestral sequences seem to be representing a transitional stage between RNA-dependent RNA polymerase and modern RNA-dependent DNA polymerase, because in a few aspects they are more like one, and then in other aspects they're more like the other, which makes a lot of sense since they seem to be a transitional stage. So in a matter of like folding and stuff like that, we think that the different outcomes of RMSD and TM score, they seem to point to the idea that the folds and the secondary structures were probably already there, already present, but they were not yet accommodated in the complete structure, which may lower the values of these parameters. But it does seem like they were already in a process of accommodation. This image here, which is the last image that I'll be showing you guys, is not a phylogenetic tree, although it does seem like it. It is actually a structural distance tree, as I mentioned when I was talking about the methodology. This tree is made from the atom distance between the proteins. So we compare these little arrows here. They represent our ancestral sequences. And we actually compared how structurally similar they are to both RNA-dependent RNA polymerases, which are the blue ones, and the RNA-dependent DNA polymerases, which are the red ones. So as you can see here, they seem to be closer to the RNA-dependent RNA polymerases, which means that in a structural level, they are more similar to RNA-dependent RNA polymerases. So as you can see, as we compare more, as we use more data, as we analyze other aspects, there seems to be a consistent pattern of suggesting the RNA-dependent DNA polymerases came from RNA-dependent RNA polymerase ancestor. So some final considerations or some take-home messages. The transition from RNA to DNA is one of the major evolutionary processes in the history of life. And I saw a question and I got distracted. It is a consensus in the scientific community that RNA was the first informational molecule. We don't know if RNA was alone or if it had the proteins, but it was the first informational molecule that much is fixed. And we also know that in many cellular lineages, mainly living lineages actually, except for the RNA viruses, DNA seems to be the main thing. And then because of that, in this work, we decided to focus on proteins that could transfer the information from RNA to DNA. And then we focused on RNA-dependent DNA polymerases, since they have both the RNA-dependent DNA polymerase activity and also DNA-dependent DNA polymerase activity. It's a lot of words, it can get confusing at times. And when we analyzed our in silico predictions, we came to the conclusion that probably the RNA-dependent DNA polymerases They come from a series of modifications in ancestral RNA-dependent RNA polymerase. As this is also suggested in that other article that I showed you guys, which is other data from our study group, which suggested that DNA polymerase of the family B also came from RNA-dependent RNA polymerases. So this data set for this large group of proteins allow us to construct a comprehensive picture of the events that may have occurred in the early stages of biological systems. Yeah, I guess that's it. Thank you so much for listening. This talk was mainly based on this article here that came out recently. This is Savio's emails. He was the corresponding author for that article, in case anyone wants to talk to him. In case anyone wants to talk to me, this is my email. As I mentioned, I'm an undergraduate student at Universidade Federal da Paraíba. So thanks to the university for the structure. Thanks to NoRCELVI for giving me a chance to talk a little bit. I have been in NoRCELVI events for a few years now. I have a NoRCELVI pin that you can actually see. Thanks to the Evolutionary Genetics Lab. Thanks to Savio for supervising me for the past few years, or a year and a half.
Well,
And
first
thanks
of
to
all, it's
CNPq
a real pleasure
for
to be able
financing my
to
scholarship.
talk with you all today, this evening. It's a great opportunity for me. I'm not going to talk to you about any chemistry or biology. Jose's given us some wonderful biogenetic work there. What I'm going to talk about is a little bit different. There's probably not a lot of science in what I'm going to say. It's really a slightly more philosophical take, but really goes to the heart of how we might think about the concept of life. So with that in mind, the title that I picked for this is Life as Process. So hopefully that will make a little bit more sense as we go through. So one thing that I've been interested in over a few years now is the idea of how our perceptions about life, in particular how our perceptions about the concept of life, how that might change the way we understand it and the way we try to build scientific experiments around it, and perhaps even ask different questions about it. So that's the kind of thing I'm going to look at today. And hopefully I'll give you a little bit of a spoiler as to where I'm going to go with it. I'm going to talk about things called boundary conditions, which are pretty common in most sciences, but I'm going to talk to you about them specifically as they apply to this concept. So, how can our perceptions about the concept of life limit our understanding or change our understanding? One of the ways in which it does that, and perhaps one of the ways that it's a problem, is we quite often phrase the question as it is shown here, like, what is life? That's a bit of a problem, really, because the verb that is underlying is the verb to be, which relates to a state of existence of something. And that state of existence, there's a few examples shown in the image there, that state of existence is said to be built of a number of behaviors, characteristics, properties, abilities, or qualia, whatever they're called. But there are certain characteristics. Sometimes this can be helpful, especially when we're talking about things like life. I could have picked any different model, scientific model of the concept of life. I just picked this one by Koshland, which is his 7 pillars of life. What Koshland has done here is he's picked out a number of characteristics that all living systems tend to have. They're listed here: program, improvisation, compartmentalization, energy, regeneration, adaptability, and seclusion. And this is given the acronym PICERAS. Now, the interesting thing about this, and I think this is a wonderful idea, a wonderful way to frame the concept of life. But if you scratch each of those different terms in turn, you'll find that each of them pertains to a process of change. And you can go through the paper and you can cut out the text that Koshland uses for each of these, and you can see that each of them relates to a process of change. So that sort of begs the question, what actually is it that's doing the changing? Now, the way we usually frame this as scientists is we have a system. We conceptualize a system, and we say the system is the thing that is changing. Now, a system could be a biological organism, it could be a mountain, it could be a river, it could be a thought, it could be anything. But what we do is we separate it from what it is not. So the system is different from what we call the surroundings. But both system and surroundings are concepts. So the question then is, how are we separating them, and what is the separation meant to be doing? Well, the reason we separate stuff into concepts is because that allows us, through our minds, to make sense of things. And quite often, the concepts are used to understand other concepts. So every concept is built through the interactions between other smaller concepts, if you like. So, for example, in this little picture here, we have happiness in the middle, the big concept, and then around the side, we've got a number of smaller concepts which represent all those things which need to interact together in order to achieve the central concept. Okay. The key thing I want to emphasize here is that what we're talking about is interactions. So we understand concepts through the interconnections between other concepts. Okay. And in order to understand a concept such as life, for example, we try to build, as far as we can, through different methods, interconnection patterns or patterns, networks of connectivity around other smaller concepts, maybe scientific ones like RNA, DNA, maybe slightly different concepts around populations of biological cells, for example. But those interconnections allow us to understand a larger concept. Another way of thinking of this is, here's a picture of a dog. If we want to understand the picture of a dog as a whole, we need to piece together, in this case, it's a jigsaw. So we piece together the individual parts, the individual concepts, to achieve an understanding of the whole. So the whole concept is understood through interconnections between smaller component concepts. And there's all sorts of different ways in which we can build those connections between concepts. So we can use human language, for example. We can use the medium of art. We can use mathematics, which is quite often the interconnections that we use through the laws of science, or we can use algorithms, for example. And I'll mention algorithms a little bit later. So, having set up the idea of concepts as being foundational to our understanding of life, and interactions even more foundational to our understanding of concepts, what concepts lie at the heart of our sort of Western scientific view about life, and are they useful to us? That's the question I'm asking here. So when we think about some of the fundamental Western scientific views about the concept of life, what propositions or assumptions are fundamental to them? I'm going to highlight three here. One, that life is a property of a thing. Second, that life is the property of a biological thing, so an organism, for example. And the third one is that things can either be living or non-living. There is a binary distinction between what is living and what is non-living, what has the quality of liveness and what does not. So, for example, when we talk about things as being alive, we might talk about a tree, a dolphin, an amoeba, whatever it is. The thing about those is that at the heart, what is underneath all of these objects, whether they're animate biological or inanimate, non-biological, is that they are all the manifestations of collections of more fundamental interactions. So, for example, bacteria and biological organisms are built of the whole suite of chemical reactions that go to make up their internal composition. We can say the same thing about fundamental subatomic particles. Their existence as things, as objects, only manifests through interactions. Same can be said about thoughts and cognitive ideas. It's only through the interactions of neuronal activity and electrical activity that we generate these concepts. And the same is true of emotions. Interaction lies at the heart of all manifest things. We could argue, and this is a sort of fundamental premise, that when we think of life, we think of biological things, but realistically, whether or not that's a tree, maybe an inanimate object is an example of a non-biological thing, but we have chosen to privilege biological processes over non-biological processes, and we call one set living and another set non-living. The real question is, whereabouts does that selection of biology being live, whereabouts does that exist outside of the human mind? That's an open question for you. The third thing, and the third property, is that living things can either be classed as living or non-living. There is a binary divide. Humans we might consider to be living, whereas a mountain we would consider maybe not to be living. But the question here is that what we're doing is we're creating, and very logically creating, boundary conditions. And those boundary conditions allow us to pinpoint differences between different concepts, the concept of a human, the concept of a mountain. The one thing that is fundamental to everything, though, and it doesn't matter if something is alive or not alive, you think of a biological organism or an inanimate object, processes and interactions are always there. They never go away. So by focusing on the processes and the interaction, you're actually focusing on something that is fundamentally always present. So what I'm now going to do is I'm now going to bring in the concept of boundary conditions. Those three underlying principles that we have about the idea of life, that life is the property of an object, life is the property of a biological object, and life is in some way binary. There is a distinction between living and non-living. Those three things are examples of boundary conditions. And there's a great paper by Daniel Paxi from 2011 that can give you a little bit more background to the idea of boundary conditions as they apply to science and as they apply to many other areas. Those three implicit propositions, they are in effect, they're boundary conditions on our idea of life. So number one, that life is associated with things or organisms or systems. That is a structural boundary condition. So for example, we can identify a structural boundary, for example, a house, and then we can then say if something is either outside of that boundary or within that boundary. So we can make a distinction. That's an example of a structural boundary. The second boundary, that life is really the province of biological things, really pertains to what goes on within the system, within the structural boundary, what sort of mechanisms of process are taking place. So in biology, for example, we talk about chemical reactions, metabolic, interconnected networks of chemical reactions. But that's just another way of saying that the mechanistic boundaries are associated with the processes that take place, such as playing a game of cards is another example of a mechanistic boundary condition. There are limitations to the way that things interconnect in order for the system to manifest itself. In this case, the game of cards. That's a mechanistic boundary condition. The third boundary condition is a little bit more tricky to get one's head around, but here's just an example. So the idea of things being alive or not alive is in effect setting up a conceptual boundary. So, for example, here's another example. This we might consider, this is a piece of art by the artist Stephen Conroy. Here's another piece of art by Jenny Saville. There's a group of bricks. Maybe some people might consider that art, maybe not everybody. But the question is, what is it that allows us to identify the picture of the woman and the abstract art as examples of art, and yet the pile of bricks is not art? That's a conceptual boundary that we decide upon. There is no fundamental, objective, conceptual boundary that separates art from not art. So those three boundary conditions, structural, mechanistic, and conceptual, are really the things that underlie our ideas about the concept of life. And when we think about life in a systems context, and here's just a little picture of a flow diagram of a system that we might identify. It doesn't really matter what the system is, but we identify components that are connected in some way. There's the concept interconnection. They're connected in some way so that their activities are coordinated. So what we have here is we have the idea that systems are inherently bounded. And if we're to understand how systems interconnect, then we need to have a little bit of a closer look at the boundary conditions. And in particular, what I want to emphasize here is that when we think about the concept of life, what we're really talking about is how those boundary conditions change, the processes that allow those boundary conditions to change.
5 minutes left.
5
No, 10. Sorry, 10
10.
minutes left.
Perfect. That's just right. Just what we need.
Great.
So here's a simple example. Here's a concept of there's a picture, concept of a tree. We can look at anything associated with that concept that might change. So we can change the structure of the tree. Its leaves might fall off in the autumn. That's a change of form, change of structure. Mechanistically, the tree changes over time as well. Branches die off, new shoots and branches form. The mechanisms, the fundamental mechanisms that allow that to happen, they change. The fundamental mechanisms that allow the tree and the branches and the leaves to move also change. But also the tree changes in concept. How is that possible? Ultimately, through the processes of interconnection or interaction. Because we might think of the tree as being conceptually fixed. It's a tree. But what do we mean by that? Does the concept tree include the concept leaf? Does it include the concept of leaf movement? Does it include the concept of roots? Does it include the concept of wind blowing through the leaves or water interacting with the roots? There's a whole suite of different interconnections that work with the concept tree. Whatever is changing, whatever allows the system to change, is therefore going to be part of a changing concept. And that changing concept is, in effect, is the process of livingness that underpins any system. Whether it's an individual tree or whether it's an ecosystem, whether it's an individual biological organism or a population of organisms, as we saw in Jose's presentation just before. So what I've done here, and I realize we've only got about 5 minutes, I've just reformulated the 3 propositions that we started with. So instead of life being the property of an object, and that property is of a biological object, and that there is a binary divide, what I've now reformulated them in, and this is a different way of parsing, of chopping up the meaning of the concept life. I'm now looking at it as though life emerges from the dynamic interactions between whatever actors are present, whether they're material, they're cognate or sensorial. And those actors and interactions have no fixed structural, mechanistic, or conceptual boundaries. We set them. And then if we want to understand what livingness is happening within those boundary conditions, we need to integrate all recursive processes of interaction. By recursive, I mean processes that interact on many different levels. We integrate all of those over the whole of the boundary conditions. And there's all sorts of different ways in which we can play that process. I'm just going to give you one brief example for a minute. If we look at this example here, here's a picture of a garden. There's all sorts of things we can see in that garden that we might consider as being alive or living. So I would say, what processes of livingness could we identify in this image? I'm sure we could all see, if there were animals in there, we could imagine animals, we could imagine plants. What we might not initially consider, though, is every process by which any individual animal or plant interacts with each other. We might not also see every interaction between every object in there with the wind, the soil, the water that is passing through that system. That is something we might not necessarily consider. And equally, every interactive process that is taking place with us now looking at that image, that is also a process of interaction. And they're all together integrated form a new place in that system. Now, in the last minute, I just want to emphasize that what I'm talking about here, I'm not making a distinction between biological interactions and non-biological interactions. No distinction there. I'm also not making a distinction about what happens on the surface of the image and what happens underneath it. So, in effect, what we're talking about is life as a recursive suite of interconnected processes. This plays out in the physical world. For example, Matryoshka dolls are an example of recursive physicality. In mathematics, we're familiar with things like the Koch curves, which give rise to fractal patternations. That's a mathematical form of the same process. And there's all sorts of different ways in which we can use mathematics or algorithms to represent how the processes of interactions can integrate together. This is not a new idea. It's most certainly not my idea. People have been looking at this for ages. Here's just a few examples. Roger Penrose has developed mathematical models where you can fit two-dimensional shapes in what are called aperiodic crystals to fill a two-dimensional landscape without repeat. And this is effectively an algorithmical model in 2 dimensions. Same thing happens in the area of cellular automata. For example, Conway's Game of Life is a wonderful example. It's a real example of livingness. And Stephen Wolfram, for example, has built a whole idea of physics based on algorithmic interactions. And these things apply to, for example, biological populations. So that brings me pretty much to the end. Most of what I've talked about is some ideas that have been, I've been working on with a number of colleagues. Kuhan Jandru, of course, is here somewhere in the audience. He and I have been talking about this for some time. And Sohan is also aware of a meeting that we held as part of the International Astronomical Union back in April of this year at Durham University. And the proceedings of that have a couple of papers that myself and a colleague, James McCrum, from St Andrews University, have been working on. The first of those papers talks about the boundary conditions that I've been discussing with you today. The second one actually goes on to show how you can use algorithms to map these ideas of boundary condition change to broaden the concept of what we consider to be livingness, and what we consider to be alive. So I'll let you, there's a nice little picture of James. That'll be coming out, hopefully, I think in December. You'll be able to have a look at that. I think the Conway paper is actually going to be on archive in the next few days. You'll be able to have a look at that. That brings me to the end. Hopefully, I haven't overrun the time. Just to thank a number of people who have collaborated with me on this over the past few years, and of course to NoRCEL for their continued support of what we've been doing here in Leeds in this space, and you this afternoon or this evening for your attention. It's been a real pleasure.
Good morning or
Thank
good
you very
afternoon,
much.
depending on your time zone. Anyways, I hoping all of you are having a wonderful day today. And we are ANEL SANJUAN and Eduardo Fonseca, and the present work was made under the tutoring of Dr. Alejandro Heredia and supported by Dr. Alicia Negron and Master in Science Alejandro Paredes in the Chemical Evolution Laboratory at the Nuclear Science Institute, ICN. Our team's research is focused on chemical evolution, and today we will discuss our most recent work on the hydrated electron. The objective of this work is to present a valuable insight in the hydrated electron stability under the cavity model. So I leave you with my partner.
Okay. So, over 2 centuries ago, the scientist Humphrey Davy noted the appearance of potassium crystals in ammonia vapor, describing them as possessing a beautiful metallic appearance and a fine blue color. Today, questions about this topic are not very clear yet, and our team and I would like to explore a new perspective. Even though the Humphrey Davy observed the hydrogen electron 200 years ago, it was proved the existence of this species until 1962. Okay, so what's the hydrogen electron? Today, after many experiments, research, and computational simulations, we know that the light absorption that produced the metallic appearance mentioned before was caused by the existence of a species called the hydrogen electron. The energy causing this effect could come from different sources, many different sources, such as X or gamma rays. We have also probably visible light, sonolysis, etc. The result of the interaction of water with these types of energy gives as a result the hydrogen electron. I put here one of the models that we are using in our simulations. So the result is an arrangement. It's like a cage of molecules of water, and in the center of this arrangement we have what we are calling the hydrogen electron.
Well, the hydrated electron is a type of solvated electron, thus meaning an electron can exist in other solvent molecules, but because it is in the middle of molecules of water, it is called a hydrated electron. This species is highly reactive, electrically neutral in liquid water, has a negative charge, and acts as a reducing agent. They can be produced by pulse radiolysis, ionizing water molecules by X-rays or gamma rays, or even by plasma interactions. Well, the lifespan of a hydrated electron depends very much on the conditions of the reaction, but it is extremely short. For instance, its lifetime in pure water at room temperature, it's approximately about to the power of -3 seconds. This is one of the main reasons because of why the hydrated electron is so hard to study. Well, the most accepted model to describe the hydrated electron structure is the cavity model, and as Eduardo mentioned earlier, it occupies a spherical hollow space surrounded by orientated molecules of water. In that sense, this model is not restrained to a certain structure or a distribution of the cavity. And well, at this point, it's already clear that the hydrated electron is not a reactive oxygen species, but the link between the oxygen and the hydrated electron is evident.
So why study this reactive oxygen species? There's an ongoing debate about its structure, ways of stabilization, dynamics, and energetics. Even though it was proved in the 1960s, there are a lot of things that we don't know about this species yet. It is very relevant in chemistry radiation. As I mentioned earlier, when you irradiate gamma rays with water with gamma rays, best say it, the formation of free radicals causes DNA damage. So there's another field of study there. It has environmental applications due to its high reductive power. This species has started to be used to treat some pollutants in water. And last but not least, we have the possible link between the hydrated electron and the emergence of life. I said it before, and I'll also say it, but the energy that causes the existence of hydrated electron could come from different sources and results that primitive Earth has a lot of these types of energy. These types of energy could come from the high amount of asteroid impacts that we are thinking that the Earth possibly had in the past. The UV radiation, high energy particles in the form of cosmic radiations, and geological activity. As we know, the continents are in constant movement. So this energy released by the movement of these great masses of crust in Earth, in the planet, could release sufficient energy to create a hydrated electron. So considering all of these energy sources, which come from everywhere, the existence of hydrated electron in primitive Earth does not sound crazy. Their interaction, the interaction between hydrated electron with matter, specifically with organic matter, could result in the creation of some organic molecules that are very relevant for the existence of life.
Well, we use the software HyperChem to model our clusters, and we basically based on our work in two levels of theory. That's MM plus, and well, it stands for molecular mechanics, and it's a classical mechanical model, and PM3, a quantum mechanics semi-empirical model. Well, returning to the cavity model, the simulations were made under this model. And at this point, we can imagine our clusters like grapevines, the vines being the clusters. And we simulated clusters ranging from 4 to 10 water molecules enclosing a free electron. Well, geometry optimization in MM Plus was the very first step in every simulation to ensure a better organization of the molecules. And the hydrogens were forced to toward. No, sorry, the oxygens kind of forced to point towards the center. I want you to remember that the cavity model does not require a specific arrangement or orientation of the water molecules. Well, the parameters that we considered for this project were the total energy and dipole moment in both theory levels, and the binding energy and heat of formation for PM3, or with PM3.
So here I'm showing the results that we have on MM+. They're not quite reliable because, as mentioned, ANEL, This model is based on classical mechanics. So if you see, there's not we can see nothing clearly, but these results are essential from passing to PM3, that is a quantum model, and it's more complex. The energy of this model is all over the place with a clear tendency in the behavior of the electron. But if we see the PM3 model, we have something that is very interesting.
Well, for understanding the stability of a system, the total energy, the binding energy, and the heat of formation, less energy means more stability in the system. Well, negative values of, for example, negative values of formation of energy indicate that energy is released during the formation process, which generally correlates with a higher stability. And as we can see in the graphic, these results are kind of linear. It follows a pattern. And the tendency indicates that the number of water molecules increase. When the water molecules increases, there is a decrease in the energy levels, and thus we can understand that the most water molecules, the most stability the hydrated electron shows.
Okay, so we pass to the dipole moment results. I am showing both of the results with both models, MM plus and PM3. Again, in MM over MM plus, we don't see a clear tendency, but in PM3, we see that the more number of molecules that we have, the more bigger the dipole moment becomes. So there's also a connection between the values of the dipole moment and the stability of the arrangement. The more great, the more big that are is the dipole moment, the more stability that has the arrangement. Okay, so here are our conclusions. The tendencies of the energy and dipole moment values point to a system's behavior becoming more and more stabilized. The more water molecules in the arrangement, the more stable it becomes. To achieve conclusive results for this behavior, more simulations involving an increasing number of water molecules are needed. Could 100 water molecules reach a higher stability? We don't know yet, but in the future we hope to do more simulations to answer to these questions. I think that's all. We have the reference here. I don't know if someone has.
Well, or morning to all of you. So I forgot to put in my resume that I'm from the Planetary Science and Astrobiology group in our university, and I come here to show you. Actually, it's a compilation of many proposals from a bottom-up perspective, leading into some aspects of life. Of course, we'll see what we will find in the road. So, well, actually, I have this huge model, which is trying to be like a map. So many of you as researchers and your work can be put somewhere around here. So we will start, as always, in the beginning. So when we are looking, all of astrobiologists, we are working in this compositional aspect of life, which is an abundance of chunks, atoms, and actually an abundance of atoms between iron and hydrogen, which are the medium size atoms that make all living cells, all living beings, and rocky planets. So my proposal will be centered around this, around life as we know it, around the possibility of life emerging in a system similar to Earth, which is a rocky world with water. So we'll see what we will find. So we have our 4 fundamental forces in the universe acting within each other. So there is this proposal from Popper, who makes like these classifications of fundamental properties, physicochemical fundamental properties, and some other properties derived from this first one. The importance here is that all of these properties act upon matter and upon the structures, the dissipative structures that can be formed. So if we go from astrochemistry to geochemistry, we will find this amazing system, which is a dissipative structure like our planet. So when you have a mineral that is in touch with liquid water, you have an interface. In this interface, you form many gradients depending on composition and many other physicochemical properties. And these ones also create fluxes, and all of the later, the before, can be acting cyclically. So in young Earth and in other systems similar, we can find 3 types of gradients. So we have this thermic gradient, and somebody early in the morning talked about ionization radiation decay, which is one of the sources of thermic how do you say? Thermal energy from the core of the planet. And of course, you have the energy coming from the sun. You have mechanical gradients, which can be from altitude in geomorphology or in pressure. We have in geomorphology, we have like this potential kinetic energy, and we have pressures in our columns of fluids. We have atmospheric, oceanic fluids, so you know there is different pressure between them. And we have, of course, gradients in concentrations from salts and ions, everything in a liquid system. And we have dynamic events between all of this. So these ones give us the main scenarios for origin of life, which are, of course, the places where we have hydrothermal activity. So we have in surface, we have some events, mostly linked to, of course, hot springs, and what happens when I have a dynamic flow of dry and wet cycles. So, of course, there is the proposal of Damer and Dimmer, some their last names, which explains really good within of these cycles and dry and wet, how we can accumulate molecules, how they can ensemble and polymerize, and many evolutionary events can occur. Oh, I'm so sorry, I started and I didn't say the most important thing, sorry. So all of the story that we are looking through from the astrochemistry, from the geochemistry, now enters the door when we arrive into this hydrothermal sites to these evolutionary domains, we will call them abiotic, prebiotic, protobiotic, and biotic. So these are not definitive closures, there are gradients. All of the processes that happened before are happening in everything that happens in the abiotic world happens in the biotic world, but new possibilities emerge. So we are trying to see how these networks start to give rise to new kinds of systems, following the same rules, following from the 4 fundamental laws of physics in the universe, what everything comes up to. So this is our dissipation hierarchy. So now we can continue. And the other side, that it's of course the rock star of origin of life, most of all from a metabolic point of view, metabolic first theories, which are the hydrothermal beds, alkaline white hydrothermal beds, which has a beautiful engine of many mineral surfaces, offering chirality dynamics, offering this network, labyrinthic network of pores where you can accumulate, and many biomolecules can interact with each other. We have this really hot, really cold, really acid oceanic water coming into contact with this really hot, really reducing environment that is coming from the tectonic activity. So it's amazing, and many, of course, I know many authors have hypothesized many aspects of life emerging in these minerals. So the main important thing for me is that we have a huge gradient of pH and a huge gradient in temperature, and now we can focus in this one of the transversal phenomena, which we can see that is thermodynamics. So we know for each reaction to happen, we have to acquire the reactants have to acquire certain level of energy so they can transform into products. So our minerals allow us to make this catalysis. Everything goes around catalysis. Our enzymes in biologic world make that, but of course the minerals can do this in many combinations. They can create a very diverse system of catalytic networks. Also the redox potential strategies like electron bifurcation, all of this has to be taken into account. Well, anyways, so we just imagine we have our molecules are evolving here. We have some related to informational aspect of life, some related to the membrane and encapsulating systems that we need, some phosphorylations, and so on. Everything can happen here. And if we make the jump from abiotic to prebiotic, to protobiotic chemistry, we arrive at this proposal scenario. Of course, well, life could emerge. We have many proposals. You can see some familiar names over here: Horowitz, Miller, Lascano. Maybe all of them could be happening, co-evolving. And we have also like this really important ring from the metabolic point of view, which is the RTCA, sorry. But anyway, so we have seen what happens into the physical-chemical aspect. So we will see now these molecules on all the story, how they interact with each other, because this also has been modeled. So we all have this idea, no? Continuously we start for a simpler, smaller set, and they start to evolve to interact within each other. The interactions can be of many types, and we can have a universe of this happening all at once. We can have these types of reaction, autocatalytic systems, which each turn of the system makes more possible for the system to occur, because the products of the system become the food molecules for the system to run again and again, and so on. This can happen with the lipids, this can happen with the protomolecules of protoenzymes, sorry, and we can have this with nucleic acids. The idea is that this has constraints, that of course we have many possibilities, many essays, but evolutionary dynamics will start to select the more stable ones, the kinetic more favored ones, of course, and the thermodynamic more favored ones. So at the end, we of course try to speak about no lookup. I actually think that term is quite ambiguous, but as an ancestral population, because we know this is evolution takes place at the level of population, so we have a big ancestral population of primordial cells, and they can maybe, if we have luck, start going to the top-down approaches. So I don't know how much time I have left. I wanted to go through everything. The many important things is that we have, how much do I have left of time? Sorry.
10
Yes,
minutes.
yes, 10 minutes.
Okay, thank you. So I hope that you understand I was not so messy, but the idea is I wanted to show you how other scientists, of course, everything here is being taken from another scientist, imagine and show us these evolutionary processes. So I wanted to make this integrative synthesis, in making it to this order, this dissipation hierarchy. And well, actually, we can say that from this point of view, which is just a small segment of the emergency of life studies, we have transversal phenomena, which are energy and autocatalytic systems dynamics and thermodynamics, which we can trace back to geochemistry, and we can see in living beings. So this is my map. I wanted to give very a huge special thanks to these authors. I don't know them, maybe some of you do, but I read them almost every day, and this work I wanted to present here. It's I wanted to recognize all of the colleagues' work in astrobiology field with this kind of big map. So I don't have it.
Thank you for inviting me. And today I'm going to talk about comets. Before I start, I would just like to extend my courtesy to some of my colleagues who collaborated with me on a number of studies that went into making this talk. So what of comets? I think many of you are actually not astrophysicists. So I'm going to start with a brief introduction of how the solar system looks like and where comets are in the solar system. So we have the sun and the 4 terrestrial planets. And if you zoom out, you have then the gas giant planets. This is Pluto, the one that's a little bit tilted. And this area, or this region in the solar system is called the Kuiper Belt. Kuiper Belt has millions and billions of small bodies that are, well, some of them are not so small, some of them are dwarf planets like Pluto, and some of them are similar to comets, icy small bodies. And then if you zoom out even further, there's a huge region or cloud, which is called the Oort cloud, which extends probably 20 to 30% to the distance between our sun to the nearest stars. And it also consists of a lot of small bodies that are moving around the sun, and it takes them millions of years to do so. So this is a comet. You can see here, this is the most famous comet. It's called 67P, or some people like to call it Chury because it has a complicated name. And about 10 years ago, there was a space mission in which we visited. You can't see it, but this is Pluto, and this is Arakoth. It looks like a snowman. It's an object that was found on the outskirts of the solar system. And it looks very similar to Chury, only it's quite larger than Chury. It's about, this object is about tens of kilometers, whereas Chury is only a few kilometers. So here's a comet. There should have been a nice animation here, but basically a comet is an object that contains volatiles. And when the comet comes close to the sun, these volatiles sublimate. So what you see here is the halo. The halo consists of the volatile gas and also dust that accompanies the gas. So you know the halo and the familiar tail, which consists of a gas tail and a dust tail. And in the center here, there is the actual comet, the comet nucleus that you saw before. This is another image that should have been quite remarkable. I'm sorry you can't see this. It looks like a blizzard storm. And it shows you what the environment looks like near the nucleus of a comet. It's quite remarkable. So there's lots of dusty particles that flow around, which were captured here in this nice animation that you can see. So what I'm going to talk about today is comets, and specifically comets that are assumed to be made from pebbles. So what are pebbles? We now think you saw some review talks before, and you saw that the sun forms, and around the sun there's a protoplanetary disk or a circumstellar disk, which is made up primarily of gas. It takes a few million years for this disk to dissipate, and in the meantime, planets and planetesimals and comets and everything is built up inside this disk. So the first solids that form are these small grains. You heard about that also before. They're micron-sized or even sub-micron-sized grains. And then they ever gently collide with each other and form these larger structures. And eventually they form these things that we call pebbles. The pebbles are like millimeter to centimeter sized aggregates made up of these small monomers, these small grains. And we have many evidence for that, which you can see from laboratory experiments and from observation and so on. Why is this important? First of all, let me tell you about some experiments we're doing, and I give credit to Jürgen Blum, who has been my collaborator for many years, and he makes these pebbles in his lab. So basically, the way you make these pebbles, which you can't see here, this is Nuria. She takes a powder. The powder is made from like these small grains, micron-sized grains, and she shakes the powder, and this is what you get. You get these small pebbles, just like you saw before, and you can sieve them and you can sort them out into different sizes, and you can make these pebbles from different materials. We make these pebbles from silica powder, for example. Here you can see iron oxide powder. We make them, there's a special machine that makes a powder of ice, just water ice grains. And we can make pebbles from that. And we can mix all of these things even and make pebbles from mixtures of different materials and different compositions. Why is this important? Because what people think nowadays, in the last 10 or 50 years, the paradigm is that planetesimals form by the gravitational instability. You have these swarms of pebbles that move around the sun, and sometimes over densities become gravitationally unstable and they collapse to form planetesimals, kilometer-sized planetesimals. So in this view, which at the moment we haven't verified this, it's only a theoretical concept. In this view, all comets are actually piles of pebbles. And so you could see here, you have lots of pebbles that pile up. These are the pebbles, and they stack up together, and there's some macroporosity between them. This would be important for the next slide. Now, you can't see the animation again, but I will explain. So we have these planetesimals. They consist of ice and rock, dust or rocky material, and they heat up. Why do they heat up? Because refractory material has radioactive nuclides. We've talked about it in previous presentations. And they release heat for the same reason that the Earth is hot. It's because of radiogenic heating. Also, planetesimals experience the same kind of heating. from either short-lived radionuclides like aluminum-26 or iron-60, or from long-lived radionuclides like potassium or uranium and so on. And you can imagine that you have initially a homogeneous mix of ice and dust, and then heat starts to build up. And then what happens to the ice? The ice can sublimate, becomes gas, or it can even melt and become a liquid. And then you have some differentiation going on. You have some movement of gas and liquid, what we call multi-phase flow of gas and liquid. And then you can get a differentiated structure where eventually you get the icy stuff flows to the cold surface, and you get an icy mantle overlying a rocky interior. So does this actually happen in comets? It's a question, because it's always a competition between two timescales. On the one hand, you have a radiogenic heating, so there's heat that is released internally at some rate, but also the heat also escapes because it diffuses from the center to the surface. So it's a competition between the scales at which heat is released and between the time scale that heat escapes. So a competition between the radiogenic timescale and the thermal diffusion timescale. The thermal diffusion timescale is the timescale it takes the heat to diffuse from the center to the surface. Now, so does it build up the heat? Well, it depends on the thermal timescale, of course, and whether we're talking about short-lived radionuclides or long-lived radionuclides. The thermal timescale, you can see, depends heavily on this delta R. This is the size of the planetesimal, and it depends on it quadratically. So this is why people think that comets, they should be pristine, because the comets are quite small, they're kilometer size. So kilometer size square, you get quite a small number, which means that the thermal timescale should be quite small, and therefore stuff can quickly diffuse, all the heat can quickly diffuse to the surface, and the object should cool quite efficiently. However, there is also this parameter in the denominator. This is the thermal conductivity. Now, the thermal conductivity can be extremely low. And the example that I give here is something that we use in our homes when we want to insulate our walls very well. We use this stuff that's called glass wool. So glass wool is just this glass sheets, and you can see that they're stacked up and there's porosity, there's voids between the sheets, but also on the molecular level, I mean, on the micro level, you can see that they also have this microporosity inside them. And this combination of microporosity and macroporosity make this stuff extremely, extremely good insulator because it has an extremely low thermal conductivity. It just so happens that a pile of pebbles is exactly the same, because if you think about it, these pebbles have a porosity of about 60 or 70%, and you also have the macroporosity between the pebbles, which is around 40%. So the combinations and the hierarchical structure of a pile of pebbles is such that the thermal conductivity of the pile could be, as you see here, 4 orders of magnitude lower than the thermal conductivity of the grain material itself, ice or silica or whatever. So it has an exceptionally low thermal conductivity. Then, if K is extremely low, that means that the thermal timescale can be extremely large. And then heat can build up inside comets, even though they're not large. Okay, now this isn't such a straightforward situation. Again, you're missing all the fun because I have great movies here. What you can't see here, that this is a pile of pebbles, okay? These pebbles are made from ice and silica combined. They're mixed together. And we make this pile and we let it thaw. And if you could see this movie, you could see that the entire pile just collapses on itself. Once you hit the temperature of melting, it just causes the whole thing to collapse. And what this means is, once it collapses, you get rid of all this macro porosity and also some of the micro porosity, and then K is larger by about 3 orders of magnitude. So during the evolution of a comet, if a comet gets to the point where melting can occur, everything can collapse, the K can be much larger and change the course of the evolution because the thermal timescale changes. There is also another potential way. You don't even need melting, you just need self-gravity. Because if you think about a small comet, the self-gravity of small comets doesn't produce a lot of pressure in the center of the comet. But as you go to bigger sizes, once you hit something like a 50-kilometer radius, your self-gravity pressure is so large, you can see it here in this image, that microporosity is eliminated just as a result of gravity. So in fact, the thermal time scale is not, I mean, the thermal conductivity is not even constant. It changes radially within the comets. So what do we do with all this information? Again, another animation that you cannot see. But this is my code, which I developed a long time ago with my supervisor, Dina Prialnik from Tel Aviv University. It's called the Structure and Evolution Model of Icy Bodies. And you can see here a bunch of equations, energy conservation, mass conservation, and flux equations, where actually all the magic, I mean, these are complicated equations and highly coupled. You can't possibly solve this analytically. You have to solve this with a computer and you can't even do this. Sorry. It's not working. Never mind. So it's just impossible to solve this analytically, and you have to put this in a computer. If you could see the animation, you could see the differentiation of an object, what happens to the ice and the liquid. It self-consistently solves both the mass fluxes and the heat fluxes and calculates the differentiation. You would eventually see this icy mantle overlying a rocky core, and you would see 2 parts to the rocky core. One part is hydrated and one part is anhydrous. So the idea is that you start with anhydrous rock, but once you have interaction with liquid water, the rock becomes hydrated. This is called serpentinization reactions. They're exothermal reactions, so they release heat. And this is a really powerful source of energy for the evolution, the internal evolution of such objects. And then at some point, they can even heat so much that they exude the water that they had previously embedded into their molecular structure. And then they are called dehydrated rock. They in fact return to be anhydrous. You can't see all of this, too bad. So anyway, we use this kind of code to do modeling of satellites, dwarf planets, etc. And we also use it for comets. And as I said before, the magic is in the details. In order to use it for comets, what you actually need is to put in the right constituent relations. And this is where laboratory measurements come in, and they give us the exact thermal conductivity and permeability of these materials. And then we can put this in the model and we can calculate comet evolution. So what we do is we run hundreds of
5 minutes left.
Please take into account my 5 minutes of missing time because of technical issues. So what we do is we do hundreds of these simulations where we vary the comet size between 0.5 kilometer and 50 kilometers. This is the radius. We vary the formation times, which affects how much radiogenic nucleide material you have and therefore how much they heat during the early evolution, the first few million years, and the long-term evolution as well. We vary the pebble sizes, and we vary the mineral fractions. You heard 2 talks already that talk about the maximum fraction of refractory organics in the asteroid belts, in meteorites, which is about 4%. But actually, there's now lots of evidence that in comets, the fraction of refractory organics could be between 1/3 and 1/2 of all the mass of the refractories. And this is something that people simply tend to ignore, but it's very important because only the silicate fraction contributes to radiogenic heating. And then we also arrange the permeability in the model, and then we get all these interesting results. So I don't have time to explain everything, but basically what you see here, that's important because it's the peak temperature as a function of the comet radius. You could see that as the comet increases in size, naturally, as you would expect, you also get higher peak temperatures in the comet, and also past the point that you can get melting of water. That's 273 Kelvin here. This says something about the pristine surface layer of the comet. That's the layer that stays below 70 Kelvin, and it kind of still remains kind of pristine. And I'm going to skip this one. And what I want to show in this particular slide is that comets, especially large comets, are anything but pristine. They change significantly. In this particular comet, with this particular model realization and these parameters, you can see that a 20-kilometer radius comet is sufficiently large that it reaches the point of melting. And then you see what you should have seen in the movie. Everything just collapses as a result of the appearance of liquid water. Wherever there is liquid water, the entire pebble structure collapses. And this is why you see the size diminishing here. And the temperature gets, getting more increased. And there's liquid rock interactions and chemical reactions that I'm going to talk about in a few more slides. And it also can change the spin of the comet because the moment of inertia of the comet changes, so it can actually spin faster. So there's a lot of interesting and intriguing phenomena that can occur as a result of this. And of course, the density of the comet becomes much, much larger than an ordinary comet. Because an ordinary comet is very porous, as opposed to this one. And so what do we expect to get? We expect to get some kind of differentiation out of these models, okay? Our prediction is that if you have a particularly large comet, you have liquid water flow, and then you have a rocky core which is completely devoid of water, and then you have a layer that's enriched in water. So you have a concentration of water from the bulk of the comet to a near surface layer, and closer to the colder surface, you can also have amorphous ice, which traps super and hypervolatiles. I'm going to skip this because I don't have enough time to explain that. And I'm coming to the prediction that we made.
5 minutes left.
Okay, so with this particular project, we made the prediction that comets are anything but pristine. So only really small comets, like sub-kilometer comets, would actually stay pristine. And anything larger than that, you start to differentiate stuff. You don't necessarily have liquid water, but you still have some kind of differentiation and flow of super and hypervolatiles, which go on closer to the surface, and then they become entrapped in sequentially entrapped inside the amorphous ice hosts. So our prediction is that the larger the comets, the different the composition going to be when you talk about active comets. So we asked.
Sorry, we're out of time.
Sorry?
We're low on time. We're low on time now.
Okay, so we asked. This is quite important.
I'm really sorry for that.
We asked observers, comet observers, colleagues of us, to compile a sample of comets that have both size estimates, reliable size estimates, and known composition. And it took us 1.5 years because it was pretty difficult to do. And we found that there is actually a significantly a significant statistical correlation between the size and the composition of comets, especially among CO, carbon monoxide. You can see here that the ratio of CO to water increases as you go to bigger and bigger comet radii. And we can even match this trend using our theoretical model, which you see here, all these lines. Again, no time to explain what this means exactly, but we can match this with our theoretical model, which is quite nice. So this is an astrobiology conference, so some implications. First of all, if you want to remember anything from this talk, remember that we think that comets are not truly pristine objects like people usually think. Large comets like Hale-Bopp could have unique features, could have these complex molecules that result as a result of the fact that you have these liquid rock interactions. And actually, there might be some evidence for that. We have these serpentinization reactions, which synthesize molecular hydrogen. And then if you have some more volatile materials, then you can actually synthesize more complex organic compounds, even in comets. There's a pretty new paper about possibility that comets can actually synthesize nuclear bases, which are important for RNA. And you do that by invoking certain chemical reactions. And there's this idea of panspermia, that if you have comets with liquid pools that can somehow bring rise to microbial life, even if those pools eventually freeze out, they can still transfer life from planet to planet, or even from exoplanetary system to exoplanetary system, which is quite neat, but I don't know if that's really the case. One last thing is, yesterday people talked about ROS a lot. And I was intrigued by that because I wasn't familiar with ROS, and I looked into it, and actually it could be synthesized also in serpentinization reactions, and also as a result of radioactive decay. So that's also an interesting fact that maybe somebody would like to comment on. With that, I would like to thank you for listening and apologize for all the technical issues, and open this for discussion.
And the title of my presentation is The Living Galaxy. This is also the title of a book that is going to be published hopefully at the beginning of next year. And this book has 3 authors: Eugenio, that stands for Eugene, of course, Mieli from Rome, Andrea Valli from France, and myself. So rather than bothering you with mathematics and all that, I would like to convey to you the spirit of the book and the spirit of the increasing knowledge about the galaxy that has been going on since the time of Frank Drake's first idea of the Drake equation in 1961 to nowadays. And this progress is made mathematically by virtue of a method that we dare to call the lognormal of Maccone, but of course the lognormal distribution was found by at University College London in the 1880s. So I am not claiming that I discovered anything new. The combination of these is possibly new. So, analytical approach. First of all, and this is the only mathematical slide that I'm showing you. This thing is a product. Capital Pi means multiplication. Product of what? Products of a number of random variables, and we call x sub 0 the whole lot of the product. Basically, for those who are a bit familiar with probability and statistics, this is the law of compound probability that students learn about in the first years of university courses on probability. And what I am claiming in this talk is that the probability distribution of this capital X sub 0 is given by this thing. This thing is the log normal probability distribution. So the whole of my presentation here is centered around log normals, not around Gaussians or other probability distribution. This thing is the mean value of the log normal distribution. This thing is the standard deviation of the log normal distribution. And these two parameters, mu and sigma, are the parameters that you can see in the formula of the log normal distribution. I have to be precise just by words. These two equations, mu and sigma square, are under the hypothesis that all the random variables that are making up the product are uniformly distributed. That is, we don't know what the value of this probability distribution will be, but we know that they range between capital B sub j and capital A B sub j. Good. Now, let me talk about the history of all this. I am working in SETI, the Search for Extraterrestrial Intelligence. And in SETI, we use the largest dishes existing on Earth nowadays, the largest radio telescopes, to find out whether or not any hint of an extraterrestrial civilization or just hint of an inhabited planet reached the Earth from we don't know where. Now, this research field was started back in 1959 by a seminal paper by two scientists, Cocconi and Morrison. And just 2 years after that, that is in 1961, Frank Drake, the greatest radio astronomers of SETI, really the founders of SETI, with Carl Sagan, published this equation. Capital N is the number of civilizations existing nowadays in the galaxy. Of course, we don't know about that, but we can write that as the multiplication of 7 factors. That is, the number of stars in the galaxy suitable for life. We assume that these stars belong to classes K, G, and F. We rule out other classes because, you know, either their lifetime is too short to let chemical evolution to take place, or for other reason that here I'm not going to explain. Then the second factor, N sub P, the number of planets per star in the habitable area. That is, the number of planets that revolve their own stars, but they are neither too close nor too far from the star. And this is what, you know, the Americans like to call, from a children's tale, Goldilocks zone. That is, I don't tell you about the story, but you can find it on the internet, of course. It's a book published many years ago in 1800 in England first, and then translated, brought to America. The third factor is F sub S, the factor of planet stable in the habitable area. Now, stable means that they are lucky in that they are not going to be hit by astrophysical phenomenon that would kill the life on that planet. We now know much more than Frank Drake knew in 1961. And so this is one of the factors that myself and my co-authors have been trying to investigate more in depth, mathematical depths, in the book that is coming out next year early. Next factor, oh, this is difficult, but difficult for me. but probably much more likely for many people that are attending this conference. The fourth Drake and the fifth Drake. The fourth Drake is the fraction of suitable planets where life actually develops. And we try to enrich this simple definition by adding more and more variables into the statistical Drake equation, so that we are updating the knowledge from the year 1961 to the year nowadays, 2024. The fifth Drake is the fraction of planets inhabited by intelligent life. Of course, we don't know what that is. We can only speculate statistically and mathematically. But the point is that in our book, all these different, apparently different factors are merged into a single mathematical structure based on the log-normal probability distribution. And then there are the two final factors in the Drake formulation. F sub c, the fraction of planets where intelligent life decides to communicate. This is important because what we are doing is SETI, the search for extraterrestrial intelligence. And so if those guys over there on another planet don't decide to communicate, then that is not part of SETI. And finally, the deepest question: how long does a civilization, technological civilization, last in time? At the time of Frank Drake, nobody knew about that. We don't know about that either, because as I said, we astronomers did not find any extraterrestrial civilization, but we can certainly speculate mathematically. So this is the situation as it was in 1961, and now we jump from the 7 factors to 50 factors. So here is the list of the 50 factors, but before going on with some details, because the time, of course, is not much, let me tell you that essentially what we try to do is to enrich and update the knowledge that we have about life in this galaxy to the present day, rather than to the 1961 when Drake started. So apart from all the mathematics and theorems and all that, the point that I would like to stress and convey to you is that this method that I worked out basically 15 years ago, called the log-normal method in this book, is enabling us to extend the knowledge by incorporating current knowledge into an old mathematical structure. Thank you. So I'll skip because the time is not much. But anyway, the Drake parameter 1, 2, and 3 are basically astronomical things. And we have pretty good knowledge about that nowadays. Much more complicated are the parameters Drake 3 and Drake 4, because they involve life. But this is precisely what I'm asking the attendees of this conference to hopefully think about, if they like, to see what could be added in a mathematical form based on log-normal distributions. And so just to give you some names, the multiple star system, meaning that, of course, the solar system is made only by one star, but there might be 2 stars or more. For instance, Alpha Centauri nearby us is made by 2 big stars plus a third smaller star. So gravitationally speaking, this has an important relevance on the evolution of life on that system. And then other problems, obviously. Supernova are stars that explode. And we are assuming that the nearest supernovae to the solar system are more than 40 light years away. Otherwise, we wouldn't be here at the moment. Next thing, gamma ray burst. They must be far away, more than 5,000 light years away. Super flares of the central star, this fortunately will not happen to the sun. Transit of gas giants on internal orbits. For instance, just imagine that Jupiter is moving away from its current orbit and is moving towards the sun. That, of course, disrupts the gravitational stability. And so it's one more reason to stop life on a small planet like Earth, and so on and so forth. Prolonged meteoritic bombardment, instability of the rotation axis. Can you just imagine if the rotation axis of the Earth and Mars was different from 23 degrees? My goodness, this is the case for Uranus and Neptune. So that is deleterious, of course, for life on the planets around that body. Here is one more topic that we try to deepen, and this is the absence of the carbon cycle. Now, this is more palatable, I hope, for people attending this conference, and also is the question of whether that planets where we are considering life has or not a magnetic field. Okay, now let me go back to Drake 4 and 5. Now, I have to be clear, I am not a biologist, so I don't want to talk garbage now. I am an astrophysicist, mathematical physicist. But the point is that in the group of the 3 people that wrote the book, there is a person who worked about that, and his name is Andrea Valli. Now, Andrea was a pupil of an Italian physicist, or a pupil in its own, a pupil of Enrico Fermi many years ago. And essentially, the name of this person is Mario Ageno, was Mario Ageno. Mario Ageno died in Rome in the early 1990s, and he put forward a theory for the origin of life on planets that is possibly one of the best theories that we have today to explain the origin of life on planets. Again, I am not versant in this thing, so I don't want to say anything incorrect. Anyway, when you buy the book early next year, you can deepen your knowledge about that. Drake parameter 5 is metazoa, and that is the grown-up animals in loose terms. And of course, we tried also to investigate that by virtue of my lognormal probability distributions. I don't know if we succeeded. We are open for criticism, of course. And so this is one of the topics that I leave to all of you in this conference for future investigation. Finally, the parameters, Drake 5, 6, and 7. This is, so as to say, the future. Now, please remember that what is past for humans maybe already took place in the galaxy for other civilizations. You see, we are working in SETI. SETI is the search for extraterrestrial intelligence. And my assumption, as the assumption of all astrophysicists working on SETI, is that, of course, we are not alone in the galaxy, at least. And of course, there are civilizations in the galaxy that are more advanced technologically and also philosophically, why not, than we are on Earth. So the point here to be kept in mind is that what is the future for us may have been the past for other extraterrestrial civilization. And actually, let me just tell you very, very quickly that I have been trying to find out a
5 minutes left.
Yes, thank you. Okay, I'm nearly finished. I have been trying to find out a mathematical way of establishing the level of civilization of a certain civilization in the galaxy. And I used what in information theory is called the Shannon probability of information. This has nothing to do with entropy as understood 1 century ago in the theory of gases and all that. This is a different story because it is related to information theory, and you will find in the book all I've been trying to investigate in this paper. So I would stop at this point, leaving to you the big question mark, the big question mark of what is in that book. Well, you just be patient, please. You wait until early next year, you buy the book, and then you cover me with questions and discussion, possible themes, and so on. Thank you very, very much.